We'll start out this section by discussing how the perception of color and how the perception of movement, or actually where they are located in the human brain. So in deciphering where color recognition takes place in human brains, researchers used two conditions: a screen of achromatic rectangles that were expected to elicit responses that would be contrast sensitive, such as differences in brightness or darkness or what have you. The experimental subjects saw the same screen, but the rectangles were colored, as you see in the screen here. So the first screen, again, differences in brightness, but achromatic, one color. And this one, color. In the second experiment, the rectangles were moved in one direction for 5 seconds and in another direction for 5 seconds. And when regions of activation, when the control conditions were subtracted from the experimental conditions, in the color condition, the prominent activation was medial. If you remember correctly, that's towards the midline, okay? In areas corresponding to human V4, that's visual 4 in the visual cortex. In the motion condition, the activation was more lateral. It was more towards the sides. Remember what lateral means. In areas corresponding to human MT, which is the middle temporal area. We again looked at that article in monkeys, and we've discussed this area several times. They also differed, the points differed along the dorsoventral. So here you're looking at a horizontal section with the front of the brain being here, the back of the brain being here. This is the occipital lobe. So you're looking at a kind of a flat plane right through the brain there. Okay. So when you look at a dorsoventral slice, so from the top to the bottom, which would be a coronal section, you would find that this area here lies superior to this area here, meaning that it's on top of. And again, if you remember where the MT area is in the brain, that would be on top of this area here. Both stimuli triggered a response in the primary visual cortex, and if you remember, the information goes in initially to the primary visual cortex as kind of simple signals that then are sent out elsewhere for more complex processing. So let's go over the what and where pathways, the visual pathways. Both of the streams, again, begin with V1 in the primary visual cortex here, the striate cortex. Again, these are very oversimplified versions, but enough for you to know. The where pathway, more accurately called the dorsal stream, remember that, goes through V2 to the area known as MT, the medial temporal or the middle temporal, and then to the posterior parietal cortex. So this is the where pathway here. And that's the dorsal stream, so that lies above. The ventral stream, down here, ventral being belly, being below, courses through visual area V2 and then V4, and then to the inferior temporal cortex, which is right in here. This is the what pathway, and it's associated with perception of objects and also associated with storage of long-term memory. So again, dorsal stream associated with perceptions of direction and speed. Ventral stream associated with perception of objects and is associated with long-term memory. And if you want to look at the neural correlates or neurological correlates of some of these visual illusions, so if you look at this visual illusion here, and you kind of focus on the outer blue rings, in a period of time it will appear that the slats inside the rings are actually moving. When you look at the brain activation, when doing this optical illusion, you'll find that this particular illusion is processed in an area that activates MT, middle temporal. So this gives us, so since we're sensing movement in here, then we're looking at movement, direction, the sense of where it's going, and that's in the where pathway, which includes area MT. When you look at, conversely, this here, you just stare at the green dot for about 30 seconds and then turn your attention to the gray dot. You'll see it turning a little bit purplish or bluish. And you see if instead, so, and that's what you see here actually. So you kind of you fixate on the green, and what you see here is when you look at the second, the gray sphere, you get a second blurb of color there. If they alternate these, you don't get that second blip. And this is processed in the what pathway. So you're not looking at where it's going, you're trying to figure out what it is. So this then would be processed in a visual region just ahead of V4, just anterior to V4. Now let's look at some visual deficits. In this picture here you see half of it is very brightly colored, and this half you see in shades of gray. So this is what happens when someone has achromatopsia or achromatopsia. They lose color and see rather shades of gray. Color is generally, or color correlates with brightness differences. So this is how this world might appear to someone with achromatopsia. And since we're talking about color and just simply perceiving what it is rather than where it's going, where would you envision that being processed? And if you're thinking V4, you would be exactly right. So this is part of the V4 pathway, the what pathway. So here you see an MRI of a patient with a lesion in that particular area in V4. See it here, here, and here. And here you see a couple tests given to this patient. So they asked the patient to look at this dot here, and they produced a range of hues starting down here at baseline, which should match this, and started upwards, and asked the patient to tell them when he noted a difference in color. And you can see here that when the stimulus was presented to his lower left visual field, his upper right, and his lower right, he can discern a difference between this color and this range of colors here, a pretty small difference, but noticeable to him. But when the image was presented to his upper left quadrant, upper left visual field, it took this much change in color before he was able to detect a difference. The same thing with the lower, looking at a different part of the color spectrum. This corresponds to this down here, and again, lower left, upper right, lower right, pretty discernible, pretty discrete differences. And then looking at the upper left here, it looks like he wasn't ever able to quite distinguish a difference between the hue presented and the sliding gradient of hues. So again, the difficulty or visual impairment of colors is called achromatopsia. Now another deficit that arises from the visual cortex is a deficit called achinotopsia. It's a deficit in motion perception, the loss of the ability to perceive motions. So what this woman is seeing as she's pouring her coffee is kind of like time-lapse photography. She doesn't see smooth motions. She sees it here, and then she sees it here, and then she sees it here, such that it causes her to overflow because she doesn't have a sense of how fast that coffee or tea is coming out. Now, achinotopsia as a function of lesions is pretty rare because bilateral lesions in that area are rare. Impaired movement perception could also arise from unilateral lesions, although perhaps less severe. So if you just had a lesion to one side, unilateral, you might have some deficits, but it wouldn't be as severe. TMS, so transcranial magnetic stimulation, is used to, in cases like this when we want to determine function, you can actually kind of shut down that area of the brain and see what effect it has. And in fact, that's exactly what they've done in studying achinotopsia, is used TMS, transcranial magnetic stimulation, to induce achinotopsia in healthy subjects. And the disorder has also been identified in some Alzheimer's patients. Now, this was an experiment to look at the differences between what would be a cortical lesion of the cortex. In particular, here they lesioned the visual cortex, and a lesion, a subcortical lesion. In this case, it would be a lesion of the colliculus. And if you remember from the kind of basic anatomy of the visual system, there are projections to the colliculus that are involved in vision. So initially, the rats were both trained to follow where a seed was, a sunflower seed that was held out to them, and in this test, they were trained to run down an alleyway that had vertical stripes at the end at a door, or vertical or horizontal in the other lane to receive a sunflower seed. And in this condition, the animals whose lesions were in the visual cortex were able to orient to where the seed was. However, the lesions in the colliculus, those animals were not able to orient their heads to where the seed was. Conversely, in the discrimination task, where they had to discriminate whether the door was slatted horizontally or vertically, those animals whose colliculi were lesions of the colliculus, were able to perform this relatively well. However, lesions of the visual cortex created problems for those animals. Now, blind sight is a really interesting phenomenon that occurs. Blind sight is the ability of a blind person to sense accurately a light source or other visual stimulus, even though unable to see it consciously. So they're reacting to it, they're responding to it, and if you ask them what they're seeing, they're not able to tell you that they're seeing anything. In fact, they'll say, "I'm blind, I'm not seeing anything." So rather than to get you more confused, there's a really wonderful short film clip by V.S. Ramachandran, who's really well known in the field of neuroscience, and I've put the link on this slide. I would suggest, and you could also just kind of Google blind sight, and his 9-minute video will come up, and it's a really stunning video. So it's V.S. Ramachandran. So here's the spelling of that name. Dr. Ramachandran has been a prolific writer, and he has an educational background in both the field of behavioral neurology and also visual psychophysics. So if you wanted to listen to anybody about blind sight, this would probably be your guy. He's also done a fair amount of work on the neurological correlates of phantom limb syndrome and phantom limb pain. This slide provides an example of multisensory information, and this is in the superior colliculus, and it shows where visual, auditory, and somatosensory information all converge on one area in the brain. So again, multisensory information, or integration rather, is the overlapping of common coordinate systems. These are coordinate systems here that can lead to a representation of multisensory space. Another anomaly in visual perception is a condition called synesthesia. Synesthesia is a neurological condition in which the stimulation of one sensory or cognitive pathway leads to automatic, involuntary experiences in a second cognitive pathway. People who report such experiences are known as synesthetes. The experiences are diverse. Some people see colors with music. They see letters as colors, as you see here. They see both of these together. They perceive a taste with a word. Any number of different combinations with different senses. Agnosias are failures of perception, and there are a fair number of them. This particular illustration depicts visual agnosia. This was a patient who, when he was asked to name this object, he did not come up with the correct answer. He, instead of saying combination lock, he insisted it was a telephone. When he was asked to kind of mime how that was used or what one did with that, he mimed correctly. He mimed the combination lock, but he insisted that it was a telephone. These recordings are taken from a single cell in the inferotemporal cortex. So I want to break that down for you. Inferotemporal cortex. inferior temporal cortex. That would be also the ventral temporal cortex. So inferior, if it were superior temporal cortex would be above. Temporal cortex is inferior temporal cortex. Anyway, these recordings show that the neuroactivity, that neuron seemed to specifically respond to hands, because as you continue down to, let's say, a mittened hand that was less like a hand but still a little bit like a hand, you would get a smaller response, the largest response being the full hand. And then when it went on to really look nothing like a hand, and the further away from a hand representation it was, you got no signal whatsoever. So it appears to be pretty specific for the hand. So the inferotemporal cortex, meaning the bottom temporal cortex, would be consistent with our what pathway. So as you'll recall from two of the videos that I asked you to watch, one was the two of the 3 or 4 videos I've asked you to watch for this week. One was the Charlie Rose series, in which Nancy Kenwasher from MIT was discussing some of her research, and then a shorter one with just Nancy Kenwasher at MIT discussing her research. She's done some stellar research in looking at areas in the brain that code for specific things, such as this area here that is specific for faces, and this area here that is specific for bodies. And if I remember correctly, she also had identified an area that was specific for landscapes. So you've seen this before, you saw it in the videos. Again, corresponds with the what pathway, but is very specific and continues to be reproduced in many experiments in her and other labs. Now these top 3 show lesions in a subject DF. This was a woman who, when giving a task of orienting a card in a certain direction to fit it into a slot, she was unable to orient it. So that was the matching task, is to kind of put it in the same vertical field, or horizontal field rather, as the slot was. She was unable to do that, but when asked to place it into the slot, she was able to do that. And she was also able to remember doing that, or to be able to do that again from memory. And so clearly there were impairments in the what area here. So this is a ventral pathway, and yet the dorsal pathway for her seemed to be intact. So this was, again, her cortex, a model of her cortex, of course, and these are healthy subjects. So she had lesions in exactly these areas of the healthy subjects. And the activation of these areas in healthy subjects was taken using fMRI when they were viewing objects. Now, when asked to identify a shape and form, we're able to do so even when those shapes and forms may be just somewhat different than what we're expecting. So we wouldn't expect a pink elephant, and yet we still know that that's an elephant. We do know that the plaid structure here is an apple. It could be something else, but close to an apple. And that this is a figure of a person. So object constancy is our ability to recognize an object in a variety of situations. For example, if we saw this elephant sitting on his hindquarters, we would still know he was an elephant. If we saw him sideways and perhaps even from the back, we would know he's an elephant because we'd see the ears sticking out. Likewise, the apple and the figure, we could pretty much identify what it was given a number of sets of circumstances. Now, view-dependent theories of object recognition suggest that the recognition process depends on the vantage point. Recognizing that both of these are a bicycle, one from a side view and the other aerial view, requires matching distinct sensory inputs to view-dependent representations. So there are representations in your head for a bicycle. So for the side view, we're going to select specific features. We may look at the handlebar, the seat, the wheels, and then when we look at it from above, we'll still recognize the handle, the seat, we'll recognize the pedals, and so we'll know that's a bicycle. So this process would be called view dependent, such that we're able to match these features with some construct that we already have in our head, some representation of a bicycle. Now, the view invariant approach would be that we recognize major structures, or for example, just the major axis in the horizontal and the vertical position. And it's probably true, so this would be based on, not on specific pieces, but just kind of on a grosser structure. And both are probably true, that we probably use both view invariant and view dependent processes for our visual perception. And it looks like the brain processes these independently as well. So if you look here in A, so both of these are taken from the right fusiform area. And if you look in A, here you have the new objects being presented, and then you repeat the presentation in the same view. And this is bold signaling, so you know what that is. That's using fMRI, looking at how much blood is being used to generate a response. You see it's equal when it's in the same view, or it's equal when it's in a different view. And that would be consistent with a view invariant representation. In contrast, however, in the right fusiform area, you have the presentation of the new objects. Doesn't take a whole lot of blood to the brain to know that it's the same view of the object. However, when presented with a different view, the blood flow in the right cortex, in the right fusiform area, is increased, meaning it takes a bit longer, and you're matching these to what else you know about this object. And so it's interesting that, and that would be consistent with the view dependence, so that these two are processed differentially in different brain areas. Now, your book talks about the grandmother cell, and this was interesting to me. I was listening to a researcher talk about some of their understanding of how faces are recognized, how things are recognized, and they talked about the Jennifer Aniston cell. So I'm going to kind of reference that with respect to what your book calls the grandmother cell. So a neurosurgeon named Itzhak Fried, he was operating on patients who suffer from epileptic seizures. So he noticed, they take advantage of these surgeries to do experiments that will help us understand the brain a bit better. So they, provided different pictures to the subjects and saw what happened in the brain. He noticed that a particular neuron would begin to flash many times when he showed patients a photo of Jennifer Aniston. It didn't respond when he showed other female stars, or when they were random people, animals, or places. And this Aniston cell, interestingly, was triggered in a number of patients. So, is there a particular neuron in your brain whose job is to recognize Jen or your grandmother? So your book identifies three limitations. A single cell representation of Jan or Gran would be highly susceptible to error. So it would be problematic if you just had one neuron that told you who your grandmother was and that misfired, you'd be confused. The second is that a specific cell does not explain how we perceive novel objects. So if something new comes into our area, and then how is it that we perceive that? And finally, the neuron would have to adapt over time to changes in the person, such as aging, hair color, cosmetic surgery, any number of things, and that's highly unlikely. And the researcher that was talking about this Jennifer Aniston cell said that you just simply cannot have a single neuron coding for every single person you know. So it's unlikely that it happens that way. It's more likely that the hierarchical coding hypothesis is true, which says that elementary features are combined to form recognition from line detecting cells. And remember, those line detecting cells are in V1, the striate cortex of the primary visual cortex, and then move out to cells that respond to more complex stimuli. So what's the difference between agnosia and memory loss? So when presented with a set of keys and you ask both patients, what is that? They will both respond, I have no idea. When you ask the person with agnosia to pick up the keys and then identify, they will, once they're holding the keys and feeling them and touching them, they will be able to identify that those are keys. Whereas when you ask the Alzheimer's or the person with memory loss to pick up and hold the keys, they still won't be able to tell you what they are. So that's one of the ways that you can distinction between agnosia and memory loss. Here you have some tests for patients who have agnosia following right hemisphere lesions. They have difficulty with incomplete letters tasks. So they have difficulty reading those letters. They also have difficulty identifying the drawings until they get to this point here. This is specific to right hemisphere lesions. People with left hemisphere lesions have a severe language problem, but they don't seem to have these deficits in identifying these tests. Likewise, when patients had right hemisphere lesions, they performed much worse than controls or people with left hemisphere regions in being able to distinguish that these are three hats together and that this is a stapler in a different view. These drawings are by a patient who, following a stroke, developed a condition called alexia. He found out that when he was released that he couldn't read anymore. The CAT scan showed an infarct at the left posterior cerebral artery, lesion area primarily in the occipital region of the left hemisphere, although it extended into the posterior temporal cortex, so the back of the temporal cortex or the front of the visual cortex. And along with not being able to read, when he was asked to color the different items here, even though he could not name them, he could clearly color them as separate and individual items. The matching by function test is another test for agnosia. So they tested agnostic patients with left hemisphere lesions, and those were the ones that showed impairment on this test. Patients with right-sided lesions fail to recognize objects, whereas patients with left-sided lesions can recognize objects, but they can't make functional connections. So the patients with left-sided lesions, they can recognize the objects, but they can't make a functional connection between the two. For example, they would see these two as being more similar than these two. Likewise, that these two would be more similar than these two. So it's an issue of semantics. It's an issue of understanding the relationships between things, whereas the people with right-sided lesions simply couldn't name these objects. So here's a graphic illustration of that. Visual analysis is done at this level, and then over to the right side for perceptual categorization, in other words, what is it? And then to the left side where semantic categorization takes place, in other words, where is it? What does it do? What is it for? And so lesions in the left side people would be able to identify what it is, but they wouldn't be able to identify what its function was. Whereas people with lesions here just simply wouldn't be able to recognize even what it is. Here we have examples of tests for people with category-specific agnosia. So in each condition, the subject is asked to choose from the array of 5 drawings, the one that goes best with the one in the box. So of these 5, what does this go best with? Of these 5, what does this go best with? So a patient with selective deficit for common objects would perform poorly in A, but normally in B. The reverse would be expected in a patient with category-specific deficits for living things. So these are living things, and obviously the category-specific would be the canine category. So for category-specific deficit, It could be any number of things. A patient might not identify living things. They might not identify mammals as a category, so they wouldn't be able to place mammals with mammals. It's an interesting deficit with a lot of variation. So there are two hypotheses about the organization of semantic knowledge. There's a category-based hypothesis here, and that proposes that semantic knowledge is organized according to our categories of the world, how we categorize the world. For example, a prominent division would put living things in one group, non-living in another. A property-based hypothesis, so you have category-based here, a property-based hypothesis, meaning that an image has a specific property, would differentiate by texture, by color, by shape, by where those things are found, by what they're used for. So these properties can be visual or functional. And below is a computational model that was developed by a group of researchers that kind of describes in computational terms what's going on with this processing. So prosopagnosia is a condition that actually was identified by Oliver Sacks in his book, The Man Who Mistook His Wife for a Hat, although some people are suggesting that perhaps was not true prosopagnosia. Nonetheless, this is where the lesions are identified in patients with prosopagnosia, showing that the occipital lesions account for far more in total, with temporal regions not too far behind. And the reason that these total percentages total more than 100 is because a lot of these lesion areas are overlapping, and so you might find lesions in the occipito-temporal pathway. And again, if you remember Nancy Kenwisher's data from her presentation, she showed phase selective activation in the fusiform gyrus, in the lateral occipital area, and the posterior region of the superior temporal sulcus. So you see these here. She did the controls. I think that some of those were shown in her video, where she would put faces upside down and wouldn't get the signal. So she did the necessary controls to show that this is these are actually regions that are identifying faces. This slide depicts Farah's two-process model for object recognition. Scientists say recognition can be based on two forms of analysis: holistic analysis and analysis by parts. So here's holistic and analysis by parts. And the contributions of these two systems are different for different classes of stimuli. For example, analysis by parts is absolutely essential for reading and plays a part in object recognition. Holistic analysis plays a part in object recognition and is essential for facial recognition. And so there's overlap among these. It would strike me that the holistic analysis is more like what we would call Gestalt in the psychological tradition, where the sum is greater than the whole is greater than the sum of its parts, meaning that you can parse this down, but it doesn't. You can break it up into individual components, but it doesn't really give you a face. Here's a slide showing that the ability to draw objects from memory might be spared in agnosia. This is a patient who had agnosia, who was not able to identify objects, but was able to draw a map of the United Kingdom and electric guitar without problem. Ironically or oddly, when he returned for a subsequent visit, he was not able to identify these objects from his own drawing. And here's a painting by another patient who had a pretty serious accident, and the result was that he was unable to see colors, and yet he was experimenting with painting colors. So there must be some sense of color in simply looking at brightness and depths. These pictures were drawn by a patient who had visual agnosia. This was 4 years after his stroke. The first pictures, this is of a piece of celery, and this is of an owl. That's 4 years after his stroke, and you can see 14 years after the stroke that that being able to reconstruct these from memory, the ability to do so had deteriorated pretty significantly. So this has been a rather unimaginative distillation of what was written in your book, really cutting a lot out. There's a lot of stuff there that you probably don't need to know until this is a 200 level class, and I think a lot of that information was pretty weighty and not unimportant. However, maybe best served when you move up to a higher level course with at least a stronger foundation. So know what is in this particular video and less kind of the minutia that's in the text. Also, make sure that you have taken notes from the Charlie Rose video, the Nancy Canwisher video, and the first video I did. I think for your own edification, because I think it's something that you will continue to draw upon as you move forward in the field, especially if you're interested in abnormal psychology or abnormal neurology, is to make a list of all of these agnosias and aphasias and all of those. Make a list of what they are, where they occur, and what the symptoms are. I think that having a list on hand like that will be of great service to you in the future.